Hello, everyone. Welcome everyone to today's talk. The title is Virtual Carpological Herbarium of Fruits and Seeds. It is the 32nd talk of a series targeting to promote agrobiotechnology and enhance understanding of the potential applications of agricultural products. We hope the series will inspire international scholars, researchers, farmers, and business in the agricultural field, as well as the interested public. First, let me introduce our speaker today, Dr. Lautai Wei David, who is a curator of the Hu Xiuying Herbarium in the School of Life Sciences at the Chinese University of Hong Kong. His primary research interest is plant taxonomy, especially in archiving native and rare plants by field records, herbarium specimens, SEM images, 3D models, DNA barcoding, and genomics. The Carpological collection is a part of an herbarium, and it mainly consists of the fruits and seeds of plants that cannot be fixed onto a standard herbarium sheet. The collection is significant in plant species of authentication, as these reproductive parts are unique in every species. 3D model reconstruction from a number of images could reflect the realistic morphology and general color of the fruit and seeds. The newly established database, Virtual Carpological Herbarium of Fruits and Seeds, was launched in 2022, and now it archives more than 500 3D models of local species. All the 3D models are presented with user-friendly interfaces for plant authentication, education, and conservation. The philosophy and application of the database will be discussed in this talk. Now, let's welcome Dr. Lau. Dr. Lau, please.
Thank you very much, Professor Lam's introduction. Today is really my pleasure and honor to be here, sharing with you our virtual carpological herbarium of fruit and seeds. And actually, this herbarium database is part of our Xiuying herbarium. I'm going to share with you our preparation, philosophy, implementation, and application of this database. First of all, let me share with you what is carpological herbarium. It's a part of an herbarium. The larger size fruit, seed, or other plant part cannot be fixed on a herbarium sheet, would be documented in an archive process, so that all these bigger size plant part would be a part of an herbarium. Why? Why they are important? As you all may know, flowers, fruit, or seed are reproductive part. They are crucial evidence in plant ID confirmation. Especially the edible fruit or seed have to be properly identified to avoid adulteration in food industry. Last but not least, native seed authentication is the first step of conserving native habitat and forests. Why I initiate this 3D documentation in our herbarium? In 2017, when I visit Korea, I encountered a very interesting facility. There's a chamber, consists of more than 100 cameras. When a visitor standing inside, just for a few seconds, a number of photos will be taken and then transferred to a computer for model reconstruction. After 1 to 2 days, a 3D printing model almost exactly identical to the visitor will be printed out as a visitor souvenir. It's very interesting. That inspire me. How I can bring this quite commercialized or quite applicable technique to our plant documentation. Then I begin with some review on various 3D database relating to nature. As you can see from this Sketchfab database, it documents quite a number of 3D models of plants. All these are very interesting and spectacular that may be very useful to arouse student interest in learning plants. I also visit a quite specialized database, Fish Asia. It documents quite a number of plants. Some of them have voucher specimens. They are quite interactive, showing various features of the plant with your adjusted magnification. So that it further inspires me how I can bring all this concept into our 3D documentation of our herbarium, especially for local plants. So that I had a thorough brainstorming of some new elements of this 3D plant archive in our herbarium. First of all, we are very treasure the importance of this basic information from herbarium standard voucher specimen. I have to include this part in the database. How to increase or enhance the resolution of the 3D model. Display all the faces of the specimen. Even in multi-directional, we can easily see the structure. Of course, we have to document our native species, especially from the local collection, maybe some endangered rare plant or some with ecological importance. Of course, we need to make good use of all these raw material and data and integrate them into our research. And how to transfer our knowledge to the community. That's all the things I'm thinking about how to establish our 3D plant archive. And then we go to the standard operation to establish our own SOP in these 3 stages. First of all, we have to collect specimens, create a 3D model, and create a database that are user-friendly and applicable to the community usage. First of all, regarding the specimen collection, we conduct our collection focusing on some important species, local species, taking the field record and fulfill all the GIS inventory, and also conduct a thorough plant literature review, microscopy, and document the specimen in digitized form. When we finish all these specimen-related documentation, we go into the core part. That's much more difficult to us. In the 3D model imaging and construction, there are mainly 4 forming parts, including the imaging platform, imaging condition, image analyzing software, and the editing. As you can see, this forming step consists of a number of device, tools, and conditions, so that our colleagues and I would have tried various device, lenses, camera bodies in various aperture size and shutter speed, lighting, and took us more than 1 year to fine-tune all these procedures into a more feasible and cost-effective platform. All these procedures and final results we have documented in an article. So that after this major step, and we then go to the database creation. And this database, we have to include the authentication details. And we have shown the voucher specimen record, provide keywords text for easy search. And also, of course, our 3D model can show the genuine color and structure with scale bar, and also provide instruction guide for model control. Let me show you how we operate this. This is our 3D database interface. As you can see, the user can freely move the 3D model like this. And And this display can be easily shown on desktop, on cell phone, Android or Apple device. All these are easily compatible. As you can see, when we make good use of the mouse, we can easily amplify the structure and inspect. This is a fruit open with some winged seed inside, but we want to see various aspects. We can easily share this image with our co-workers, researchers, and also teachers and students for learning this species. And also we have some keyword text so that when pressing this small button, all the fruit with well-established models can be shown and easily found from this tagging. So that we have some tagging keywords. All these provide a very user-friendly approach. Even for primary students, they can easily operate this database. After we have created this database, we have documented a number of novel components of our native species. First of all, as I mentioned before, we have included a vulture specimen like this. It's a very important species evidence and documentation. We have kept this permanently in our herbarium for coming 50 or maybe 100 years. Okay, let me show you some other novel component of our 3D native species model. First of all, the magnification. This is a seed of Cocculus orbiculatus. As you can see, this seed, when we inspect this seed on our hand, it's only about 2 to 3 millimeter. It's really tiny. We have to inspect it using various dissecting mice, but if we document it in a 3D model, you can easily see every part in a very high resolution, so that we have a small breakthrough. I want to say we can document the small size object down to 2 millimeters, and we have a very wide range of documentation from 2 millimeters to 50 centimeters, so that we can document a very large size fruit or very small size seed that can be easily shared with all the students. Even if we have laboratory demonstration, we don't have to collect more than 100 seeds for display or for demonstration. We just provide them a link so that the students using the cell phone or iPad, they can easily inspect the seed structure. That's the first novel component of our 3D model. And then the next two is the complicated structure and genuine color. Let me show you this is a 3D model of Antidesma bonidus. As you can see, it's about 2 centimeters, but the genuine color, we took the photo of it fresh collected states. As you can see, the color changes of the fruit from raw to mature can be easily seen. Even after years, this model can be kept, can keep the color very clear to the students. Let me tell you, this specimen still being kept in our herbarium, but the color has become gray to brown in color, not red anymore. But this 3D model can tell the student, okay, the fresh state color and the arrangement of the fruit inflorescence will be like this. So that the genuine color and structure is the second interesting things I want to share with you. And the last thing is the native and rare plant. And when we go to the field, we have special interest in looking at the rare plant. For example, this Camellia hongkongensis, it is actually protected and controlled by a Hong Kong audience 96. It's a quite rare plant, so that when we need to protect this species, propagate this in the wild and collect the wild seed, and also to estimate the natural distribution, first of all, we need to identify the fruit and seed, so that this documentation of the genuine color, shape, and structure can help us and other ecologists to well identify this plant even under a forest in the natural environment. So that documenting a rare and endangered plant is another important feature of our 3D model database. This for novel component is our major features of this publication and database. In addition to the native species, we also have interest to document some agricultural species. This Long Wong Yat Ho is a very spectacular species researched by Professor Lam, so that we have the interest and try to document it and see how good it is. Of course, this species is also very interesting and attractive. As you can see from this 3D model of the seed. We can easily share it with our co-workers who have interest in the seed, at least if they have to select the purity of the seed mixture of a big package of seed. They want to identify the seed structure, no matter from which angle. They can make good use of this standard 3D model image to identify the seed. So that even in other locations and other regions, you don't have to send the sample to your coworker, just share the link. And also the seed pod, I mean the legume, we can easily share the structure when they are giving food. You can tell whether the species are giving the right food or seed, what you expect, so that we can amplify the structure. You can see the hairy condition on the legume surface, so that there is another kind of sharing platform. Okay, that it seems that's the end of the story, but actually it's not. What's next? We have a quite good documentation of this 3D model of native plant and some agricultural species, and also some ecologically important species. What's next? Let me show you what our next step. I would like to demonstrate how to integrate and synergize 3D model into our herbarium research and education platform. There's very important application and knowledge transfer. I want to show the example insect tree, because this species is very special because of they are native, protected, medicinal, and propagated. And first of all, we have to inspect their features, characteristics very carefully from the historical specimen, and also compare it with our newly collected specimen, including the location of collection, the flower structure, every part of this fresh and dried specimen. We have carefully integrate them into a plant fact sheet, so that we have plant fact sheet documenting more than 100 characters and the status of this plant species. After this documentation, actually we are still not very satisfactory, because most of us would like to show the structure with their descriptive terms, so that we go ahead to the scientific illustration. As you can see, we pinpoint very accurate the structure with our botanical description terms, so that all the user, even they are ecologists, they are any kind of conservationists or any biologists, they want to correlate the structure to the environment, to the animals, or to any kind of study like evolution. First of all, they're required to know the structure, how to describe it, and how to identify structure, so that we document it with this scientific illustration. As I mentioned before, flower, fruit, and seed are very important. So that we go ahead to dissect the flower. It's very interesting to us. Some structure new to us. For example, all these prominent features on the peripheral part are actually not petals. They are sepals. The real petals, 花瓣, are actually a very small part inside. So that we dissect it and show this dissection photo to the student and our co-workers and some ecologists. And also we of course, we document it in this 3D model, so that all the user, researcher can see the structure from all angles of view. As you can see, I can amplify it so that we can easily count the stamen. Actually, they have 10 stamens inside the flower. From different angles, we can see the attachment of the stamen, the rich hairs surrounding the stamen, and also the sepals, 5 sepals, and also the outside structure, which is very hairy. And then we go ahead, of course, when studying flower, we also included the pollen structure. We make good use of our SEM device to document the pollen structures, ranging from a few thousand magnification to more than 20,000 magnification, and document all this structure. And we expect all these tiny or microscopic structure will be very helpful to study the population evolution and identification. The next one, of course, we have included the fruit and seed. It is very important to study their fruit ripening stage and the seed dispersal by wasps, so that we document it with our scientific illustration and also some dissection photo. And let me show you the 3D structure of the fruit and seed. It's very helpful to let the student and our co-worker to see, oh, this is a kind of capsule. And it consists of a very thin connecting tissue of 2 mature seeds like this. And the wasp would like to feed on part of it and carry this seed for dispersion. So that all the 3D structure will be very interesting and helpful to our students and also to our co-workers. And so when we developed this database since 2009, we also established, try to transfer our knowledge to a STEAM education scheme. And this is a new scheme named has insect tree conservation on campus. So that students would visit us and take photo of the insect tree wound. And we also cooperate with them and let them try how to reconstruct the 3D model. And they can also visit us in our herbarium to inspect the specimen. And we also collect the seed. And also we have very kind donation from the AFCD, our local government unit, to provide seeding to local school so that they have plantation in our school campus. We also have some teacher workshop, field trip, and training discussion session. It's very delighted to know that since 2019, at that time, the seedling is only 3 months old. After 3 years, they become a quite mature small tree. This year, 2023, our colleagues have visited all these trees. They are successfully giving fruit with viable seed, so that we collect seed and then transfer and donate to other schools in the same community. So that all these school teachers and students, I want to share with them what we are doing is a kind of ex situ conservation scheme. They plant this rare and endangered plant out of their natural habitats and plant them in the school campus. It's not only a planting scheme, it's also a kind of STEAM activities so that the students can take this opportunity to have plantation, to try to germinate the seed. When they are growing up, they will put in some device measuring the growing condition of the insect tree, they have ecological observation assessment. Some have exercise by filling in the fact sheet and work sheet. They have laboratories studying specimen, hand-on experience on the fresh specimen. We also provide teacher training. Some senior form students also have some biological drawing and illustration training, so that all these can enrich their learning experience and also know how to protect these species by all these activities, and also execute some ex situ conservation planting. So that from my understanding, we have a new family member, 3D Plant Archive. This archive actually would not stand alone. It's a new family member of our herbarium. It really connects our research, education, and conservation from all aspects, from the field survey, specimen, GIS, rare and endangered plant, literature review, type specimen, plant anatomy, a character record, botanical illustration, pollen SEM record, barcoding, APG4 system documentation, STEAM education, and public engagement, so that it really enrich our work and help us a lot. Our current status of this 3D carpological herbarium have accumulated more than 500 mostly native species. And actually this database up to this moment included native, exotic, rare plant, urban trees, food plant, folk medicine, Chinese medicine, wheat and poisonous plant. As you can see in this database, all these are easily searched and can be manipulated or easily operated under this user-friendly platform, so that all of you or your teachers, students, and co-workers are welcome to visit this database to share your ideas with your students. And all these interesting categories are probably become our future research and education topics. And all these interesting topics and works are actually fulfill our herbarium missions to document unique herbarium specimens, drive us to our original research, to transfer our knowledge to our students, co-workers, and teachers by e-learning platform, and finally to outreach to the community. And all this, I think, is very new to us and interesting to us. All these things are well prepared and done by our herbarium staff and intern, so that I have to special thanks to our team members. They not only on this 3D archive, but also on our herbarium research and education work, and also to the community outreach. Okay, that's the end of my today's presentation. I hope you all enjoy this 3D archive. And do you have any questions or discussion, please feel free to propose and ask.
Okay, thank you, David. Like to ask question, please unmute yourself.
Hello, this is Amy.
Amy, please.
Yeah. So interesting. So a color is one of the very important properties and characteristics that let us identify the species. And but I think we all have the experience that we use different cameras, even when we take take a picture of the object at the same time. If we use different cameras, it gives us different colors. Then would there be any better ways to also document the color in the database?
Okay, let me share our experience on this. When we take picture of this plant specimen, we would take around from 100 to 500 photos. During the whole procedure, we would use a number of light pads, which has been adjusted to the wavelength around, not around, exactly 5,600K, so that this is almost identical to the white light. And that reflection would provide us the genuine color on every image, even the 500 image of that specimen. All the color are very consistent and genuinely reflect the true color. That's the really major concern of us, because that 3D model would be used for a very long time. And also we want to document the genuine color at that stage when we collect the specimen. Yeah, that's what we have done.
Wow, impressive. Thank you.
Hey, Wang Ho, do you have a questions to ask? Hey, I saw part of your question. Okay. So the question is that would the database include historical carpological samples?
Yes, because our herbarium have a very good collection since 1960s. Some of our collections are actually donated from New Asia, from Chongqing College, and from other research institute of CHK, so that we have collected a very wide range of plant specimens, including algae, some lichens, medicinal plant, some higher plant, some rare endangered species, so that our historical specimen have some their plant part that cannot be fixed on the herbarium sheet. We will put them in another archive box. In that case, we will make good use of this chance to also document that bigger size or rare or important fruit or seed in our database. Yeah, that's what we have done, and we will continue to do so.
Thank you. So I have a question. I think this is an old question that always in my mind, right? So by your database, you can investigate a lot of morphology of seeds and fruit. So we will be able to answer the question whether this kind of the shape and the morphology of the seeds and fruits are actually related to evolution, or is it a random event that we get different kinds of shape and morphology?
I think from a point of view, when answering some ecological or evolution related question, we have to document a species from various population or from various geological time period. For example, we have some specimens in 1960, 1990s, or even nowadays. When we document their 3D structure, we can see some very slightly differences in their morphology that drive us to deduce or hypothesize. This structure may be relating to some short-term changes or adaptation to the habitat or some ecological or responding to any ecological factors. But I think our 3D archive can provide a tools of documentation or records. If we want to address this kind of question, we have to collect a number of seeds from various time period and also from various populations or even from different regions of or countries, so that we can document well all these specimens, maybe a few hundred specimens of the same species from various locations. When we compare these 3D models, maybe we can find some new structure or special structure that are under the effect of the different population or geological time effect.
How about among different species? Because within the same species, you need to have lots of samples from different geographical regions. But for different species, will there be evidence to show that during the evolution is a species-specific modifications? Or?
From our record, actually we have document up to about more than 500 species. Of course, there is a trend showing that the seed or fruit under the same family, they got some character. They are identical among all these species. We also rely on these characters to identify that species to family or genus level. Sometimes we are very we have curiosity to know why this character is quite specific to that genus or family. This is still a good question, and maybe it's relating to some of the dispersal method, relation to other animals or other parasitic or any coexisting plant or animals or insects. At least I want to say this 3D model can provide the structure very clearly to let us to find out all these ecological relationship or identification structures or any ecological structures. At least we document the structure first, and then we hope we can provide more evidence and good tools of documentation for co-workers, for researchers to maybe provide us with more number of or specimen of the same species so that we can compare the 3D models. Yeah.
Okay. Any questions from the audience?
Hello, Dr. Lau. This is Sandra. Really interesting talk, and I would like to ask two questions. The first one is that currently there are many ecologists in different fields trying to use machine learning, etc., to identify to do identification of individuals from populations, or even to characterize or even to try to seek new species from an archive of museum specimens. So I'm just wondering, what is the current process? What is the current progress of such application in the field of plant sciences, and especially in plant taxonomy, etc. And also the second question is that I hope it will not be off topic. That is, in this digital age, and as you and your team have already developed a digital collection of plant specimen, then what is the how would you answer the question to the purpose of nature illustration, such as the amazing book and collection of your 香港原生植物誌? Thank you.
Okay, very interesting questions. Thank you. The first question regarding some artificial intelligence for identifying plant. I think what our herbarium is doing is to document or archiving plant characters in various ways, by illustration, by SEM, by some anatomical pictures, by 3D models, or even by DNA barcodings. I think all these data can be integrated for future identification. Even for AI, they need to make good use of some basic information. They have to learn itself, but before the machine can learn, you have to provide accurate information. a very well documented and accurate or well described database, I think is a very important prerequisite for the AI development. What I am or my team is doing is to document the characters as good as we can. Next step, maybe I don't know how far. Maybe after years, all our data can provide a very good nutrient for the AI learning and to develop the AI identification of local plants. That's what we expect in coming years. The second question is relating to botanical illustration. Actually, photos and also 3D models sometimes cannot show that a very small part or macroscopic part or even the dissected section of the fruit or seed. Botanical illustration, especially the light drawing, can show a very clear, because it only being constructed by a simple line, not in color, so that some structure that can be easily and very sophisticatedly showed up by this illustration. And also, based on this illustration, we can put the label very well and very accurate on the top of this structure, so that I think botanical illustration is a very important part of plant taxonomy. Could be a very good supplement to our identification, and also for the for learning the plant characters, and also for very important documentation. For example, let me tell you, when we dissect the plant, we usually find some new characters that have not ever been described by the florist, so that our colleagues would document it by light drawings. That's another very important purpose of these light drawings. Okay, so that I think plant taxonomy study would have many methodologies, including plant specimen collection, photo records, 3D documentation, codon records, DNA barcoding, botanical illustrations and drawings. All these all together can create a more accurate and scientific platform of taxonomy. That's what I think. Thank you.
Okay, thank you. Any other questions? Okay, all right. So I guess we don't have further questions. So just an announcement. So we will have another talk in July. So the date hasn't been decided yet, but the topic will be on regenerative agriculture. So it's a new concept. Well, not quite new, but this is a concept of just regenerative the soil by doing agriculture. It's more environmental friendly than organic farming in theory. So I think it is an interesting topic. We'll have it probably in the later part of July. So we will announce it to everyone. If you are interested, you're welcome to join our next talk on regenerative agriculture. So with that, we thank David again, and thank you for everyone to stay with us. Okay, see you and bye-bye.
Thank you. Bye-bye.